Hey, everybody. Welcome to the Growing with Fishes podcast. Apparently, I have this podcast also playing in the background in multiple places somehow. I apologize for that. Where is it still playing? We'll figure this out, guys. Don't worry. Thanks, everybody, for watching. Welcome to Growing with Fishes. Looks like it's on Kevin's end, maybe? Mine? All right. I apologize, guys. I finally figured it out. We had a weird glitch there. Thanks, everybody, for watching. I sincerely apologize. We have episode 302 today. We're going to be talking with Kevin McKernan. Thanks for joining us, Kevin. Kevin actually had an awesome paper out the other day, and I really wanted to make sure that we had a chance to touch on it. And he's one of our favorite guests to have on the show, and we're happy to have him back again today. Thanks a lot for joining us today, Kevin.
Hey, thanks for having me back. I always enjoy coming on, Steve. This is a whole biocontrol thing is, I understand, kind of dear to your heart. Good to go over some of these things.
Oh, yeah. I think a lot of people don't understand, too, when they get false positives or false negatives and things like that, and don't understand why it's happening or don't understand where the issue is. They think that they did something wrong, or they think that there's some other factor that they, falsely attribute to some of these false positives that people are seeing in the market. And you had a really great white paper out there, and I felt like it didn't get enough recognition in terms of, I didn't see it reposted a whole lot. And I wanted to make sure it really got the recognition and the education out there and in the community that people really needed to know about what's going on. So you had this wonderful paper out the other day called Biocontrol Agents and Their Influence on Cannabis Testing, on the Cannabis Testing Space. And I thought it'd be great for you to kind of break it down for us, because it was very intriguing. It talks about how Bacillus amphiquiliensis is causing false positives for things like aspergillus, and how certain aspergillus strains that are not at all cannabis positive are cannabis affecting in terms of whether or not it's actually a health factor for people that are smoking it. Can actually tell us false positive as well from other crops. So I'd love for you to tell us a little bit more about that. We have it up on the screen. We'll make sure we have a link to that in the description as well.
Sure. So the credit due here is really to Steve Cottrell and Christopher Marsh, the ones that brought this to attention. They were noticing a higher failure rate for aspergillus in Arizona. So, I think one of the lessons here is this was only noticeable because Christopher Marsh has labs in many states. And so he can see testing differences state to state that aren't necessarily obvious to us. We sell a lot of these aspergillus testing kits, but we don't necessarily have any visibility on the percent positivity state to state. So we don't have like one of these COVID dashboards that could tell us like one state being better or worse. He does in many ways, in that he's got labs in multiple states and noticed it's oddly elevated in Arizona. So he was trying to figure out what that was. So he pointed out to us one of these strains that is in frequent use down there, known as AF36. It's actually an aspergillus strain. And this is a strain, so not bacillus. We mentioned bacillus in the paper because that's another biocontrol agent that's often used in for other reasons. But in this case, people are using this aspergillus strain, AF36, often known as Afligard, to basically put onto plants. Usually it's like cotton or other plants in the neighborhood, to basically outcompete the ones that make mycotoxins. So it's a good idea, right? That if you don't want to have aspergillus that makes mycotoxins, put another aspergillus on there that doesn't make them. So this AF36 is the interesting thing about it is it's a single point mutation in the genome that makes a change. It knocks out one of the aflatoxin genes. So you're never going to be able to differentiate this with like just plating them, because you got to get down at the single base level to sort these out. And even our test that's currently in the marketplace doesn't differentiate them, because we're not looking at that gene. We're looking at a different part of the genome. We did subsequently go and make a SNP specific test that can split these if people need to, but it's regulators are going to want that for cannabis safety testing, mainly because both of these species can still give you aspergillosis. Like, they're still pathogenic to your lungs. One just doesn't make aflatoxin, the other does. So for agricultural products that aren't inhaled, it makes complete sense to use this stuff. But if it happens to be your neighbor and it's in the air for any reason, or on the ground and you're tracking it in, and you have elevated aspergillus fails, you might want to like dig a little deeper. Check your air filtering systems, and just ring us up or ring Christopher Marsh up. We have a test that's kind of an R&D here that can kind of differentiate whether it's AF36 or whether it is a wild type toxin producing aspergillus. But the reason we wrote this up is that we continue to run into this. Biocontrol agents are really good ideas. I'd much rather fight pathogenic microbes with their non-pathogenic cousins than with chemicals. And are pushing us in the chemical direction, as we have non-specific testing going on. It's like total aerobic count and total yeast and mold. We've talked about this for hours in the past, but these tests don't give us really any specificity as what the hell's on the plant. And I don't believe in the idea of growing plants in a bubble. Like, you need nitrogen fixation, you need microbes. Microbes are part of growing plants. So penalizing people for having high numbers of them is rational if you're not speciating them. So that was the impetus behind getting this kind of written up there and out there, because a lot of people may not know that there are other biocontrol agents like this in use in other geographic regions, and they might be contributing to your false positives. Or I should say your positive tests, which may not necessarily be pathogenically positive. They're positive according to a total aerobic count measurement, but I think we want to be able to bring cases like this forward to regulators and be like, look, you're putting two industries at odds here. We've got the cotton industry that wants to use this and a cannabis industry that doesn't want it anywhere near them. And they're going to start fighting if we don't come up with sensible regulations that where both parties can use these things and not have one encroach on the other.
Absolutely. And we see similar issues with E. coli. There's many species of E. coli, most of which are not human pathogenic. There's only a small percentage of them that are actually going to make you sick. But we see false positives all the time with water testing and aquaponic systems as well, in a similar manner, to where you have these things that are testing hot or pathogenic in a test kit or from a testing lab, but in function are completely not the same species or have some other attributing factor. Is there any others that you've noticed since you do so much testing? I know that many of the different mosaic viruses can, test hot as each other and other things. Is there any others out there that people should be aware of that, could potentially be a false positive, or you think that have relatives that are close that, could potentially be an issue in the future that maybe don't have documentation?
There's another aspergillus strain out there that's in use in other states under a different product name than Afligard. I have to look. I've got to dig that one up, but I'll forward that to you after this. It's escaping my memory right now. But yeah, there's another one. It has a different mutation, so it has knocked out the gene different than this G to A mutation. It's not in use in Arizona, though. It seems to be used in more the Southeast. I think they've found that these different strains work more favorably in different climates, and so they're used accordingly. The other one I've seen in a lot of products in garden stores is just the Bacillus, and however you pronounce its last name. We call it Bacillus Amy, but it's a complicated mouth twister. But that one is we see quite a bit in stores and assume it gets used to fight powdery mildew and a variety of other things, and that's going to light up your TAC counts. We know that. So that's one that is there should be more discussion about as well.
With that in mind, do you think that people should not be using that Bacillus? I'm sure I'm horribly mispronouncing it because I can't pronounce Latin to save my life.
I know that's my problem too. Is I read these things, everyone tells me you mispronounced it. I'm like, well, I've never heard anyone else say it.
Yeah, exactly right. No one else even tries to say it.
I'm a bookworm.
No, but do you suggest that people not use that, say, for instance, in flower or after, say, week 2 of flower to kind of prevent that proactively?
Well, I don't know that it's necessarily pathogenic to humans. So, I mean, this is the problem we have is we're approaching everything as like any microbe bad, and that's just not reality. It's just as many microbes in your body as there are human cells. So this is not something that we can play this hypochondriac game with. I think we need obviously more information about those, but if they're used on products that are, generally considered safe in the other agricultural food products, I don't get overly worried about them on flour. Maybe if you're vaporizing them and they don't fully sterilize it, maybe there's some, edge cases. But, if it's going through extractor, not to worry. If it's usually getting burnt, it's probably not much of a worry. But this idea of ridding them all, I think, is ludicrous. We're never going to get there. I mean, and it's very arbitrary, right? Right now, there's 100,000 counts of total aerobic counts that are allowable, right? They don't care which kind. They're just saying 100,000 that grow on this plate type, which is a whole nother like selection factor of 95% of the stuff doesn't grow on that plate. So who cares? But they still have this 100,000 number like it's scientifically meaningful. It's not, so you can have 99,000 E. coli 0157s on there. I mean, okay, that seems like a hall pass for a whole lot of pathogenic E. coli. But if you have 101,000 of this, lactobacillus you might have in yogurt and eat by the gallon, it's suddenly, not okay. So the metrics just make absolutely no sense. And I'm convinced this is something that was put in place for like 3M's benefit many years ago. It's just like, an incumbent agency or corporation has figured out how to get regulators afraid of these things. And they've put them in and rubber stamped them on every damn product going forward, including making its way into cannabis. But there's absolutely no clinical information behind this. There's no clinical reason why that threshold is dangerous on cannabis. It was never actually measured. It was just superimposed in the food industry, and they've been using them for 50 years. And the food industry, when they started using these things, had no clue what it meant either. I mean, they weren't even speciating them back then. So, I mean, people just have to recognize this plating thing goes all the way back to before we understood the structure of DNA. I mean, these things were around like in 1880. Watson and Crick didn't figure out the structure of DNA until 54, right? So this is something that has just been a legacy tool that keeps getting reinstated. And whenever you dig, there's no real like rhyme or reason as to why this was implemented. They're like, well, it's a general microbial count, and high levels of microbial count are usually bad. It's like, well, now that we can actually sequence these things and like know which ones are good and which ones are bad, it's time to like revisit this, because it's going to push agriculture in a really horrific direction, which is to just universally lower microbial counts, which may not necessarily be the right thing.
Oh, not only that, so you just touched on a whole bunch of points. So I guess first off, you also mentioned on how some of these bioagents can actually melt the petri dishes, such as the case with Bacillus amphicoiensis, which was cited in your paper.
That's a common problem with some of the microbes actually can metabolize the gels. Like the Petri gels are oftentimes made with various carbohydrates, and some of them are digestible. So this particular Bacillus one has been noted at melting 3M plates. Now, it wasn't the plastic hard part. It was the actual. If you ever use these 3M plates, there's some type of carbon source, like a gel, almost like an agarose, but it's not agarose. It's a different carbohydrate, but that they grow on. And if that thing dissolves, then your colonies all diffuse into one another. So like my background here, you can see these distinct colonies you get from DRBC. If those microbes can metabolize the gel that it's on, well, that just turns into a big soup, and you can't actually distinctly count the colonies. And that happens with that one biocontrol agent as well. And I'm sure there's a lot of other microbes that do the same thing. Such that you can oftentimes plate these things on 3M and they'll start blending together. And now it's a really subjective call. Is that one colony? Is it 2? Is it 10? Is it 100? You really, it's a very, very subjective measurement for quantitating these things.
Absolutely. And then you also touched on testing on cannabis versus veggies, which is a huge discussion as well. For example, the same, anyone that has Frontline for their dogs, for instance, that can be up to 2 parts per million on grapes and lettuce in your grocery store, but for cannabis cannot be detectable at even 1 part per billion. Okay. In California, for instance. So there's a monumental difference. And I would love for someone to take a bunch of grocery store vegetables and test them to cannabis standards and find out what
Nothing
the actual percentage of grocery store vegetables would ever pass for cannabis testing. Yes. And then let's have a rediscussion on pesticide residue testing for all crops, because I think that what we're seeing more and more, the more I learn more about cannabis, especially with people like yourself that are highly educated on a lot of these topics, is, man, if we held our vegetable and food production to even close to the standard that we held our cannabis, a lot of people's health would change significantly.
Yeah, it's very true on the glyphosate side as well. That seems to be an area where that seems to be accumulating. It's being used so heavily everywhere that I even saw a patent application recently for glyphosate resistant cannabis, which was
I saw that
shocked me. But yeah, that's an interesting threshold. The other one is, I think the mycotoxin testing is probably cranked down to 20 parts per billion as well in some states. And I've not found a lab yet that's found mycotoxin at any high frequency. Like I think it's one of the lowest failure rates in most of the labs when they tell you about what do things commonly fail for. Like, these total tests are usually at the top of the list, TAC and total yeast and mold. And then maybe E. coli, salmonella, and aspergillus are down in the 1% range. They're usually fairly low frequency, but the mycotoxins, I think, rarely ever reach levels that trigger a failure. But I've always questioned why it was set so low. And is that like equivalent to what we find in other food industries, or is the cannabis industry being, basically put to higher scrutiny due to it being an inhalation product?
Well, it makes you wonder, is it like a glyphosate or not a glyphosate, like an Eagle 20 where mycobutanol, where it converts to hydrogen cyanide?
Yeah,
is there a conversion process, or is this an actual... Yeah,
and that's a legitimate point. And that one, okay, fine. It's the pyrolytics on there are going to convert that to hydrogen cyanide. That's not what you're going to see when you eat it. So there's a totally justifiable reason for there to be a different threshold for that pesticide on cannabis than on anything else. That's actually I think that's a smart move and it's done it. I think Ethan Russo had a paper out that demonstrated that thing enriched a little bit more in extraction than cannabinoids. So, low parts per billion could easily make their way up to parts per million or 1,000 if it has a significant enrichment through extraction. But some of the pesticides do that. They seem to enrich better through extraction than the cannabinoids, and so you're going to get a difference before and after extraction.
To kind of further expand upon some of your work you've done down this field, this is another paper that you put out this year, and it's something that I also thought was really interesting and didn't really kind of get the recognition I think it needs to in the cannabis industry, is this pathogenic I'll let you pronounce it because I can't. I'm worse at it than you are.
Most people just call them entros, but Enterobacteriaceae, I think is the name, the way to pronounce it. But enterobacteria is another bit maybe easier on the tongue. But so yes, these are microbes that are typically found in mammalian gut, and they are tested for as an entero test or a coliform test. Not all states have these in place, but I think Mass has it in place, and a couple others that were following the AHP. The HP had some recommendations, and those recommendations weren't for testing thresholds. They were just, hey, this is how you might evaluate cannabis for microbial content. And they specifically spelled out in the HP, these should not be taken as thresholds for pass-fail tests, but most states ignored that and just did put them in place. Unfortunately, we have found entero and coliform tests to be instated in a lot of areas. Now, there are EB plates, which are these enterobacteria plates that 3M cells that are commonly used out there. There's also total aerobic count tests. So enteros should grow as a total aerobic count, and you should be able to pick them up, at least all the aerobic ones. We just compared, okay, if we plate these things on the two different plates and we measure their DNA and we put them at multiple different temperatures, what happens? And what we quickly found was that a lot of the microbes would only grow at one particular temperature and not another, and would grow on one plate and not the other in a very temperature dependent manner. So it's just another one of those reminders that, most of the labs are actually growing those plates at 37, which many of the microbes didn't grow at. Unless you put the temperature down to 26, they didn't show up. So if you put these microbes at one temperature, you're not really counting the microbes. You're only counting the ones that can grow at 37. And likewise, if you only use an EB plate versus a TAC plate, you're going to see some microbes on one plate versus the other. So just another reminder that this whole measurement system of colony forming units is a very subjective game. It's almost like relativity, where depending on how and where you measure it, it changes on you. So it's a bit of a Heisenberg uncertainty system. So that's very frustrating because we're always asked, hey, you have to get the same CFU. Your PCR's got to measure the same CFU. And we're like, okay, please define what temperature, what time, and what carbon source you get those CFUs, and then we can calibrate to it. But every time we try and calibrate, the gold standard keeps moving around. The goalposts shift from one state to another, saying, okay, in this state we're using Michigan likes to use DRBC, this purple stuff behind me. Some other states are okay with multiple different systems being used, and so none of them ever agree. And the labs are always into finger pointing games, accusing the others of lab shopping. I don't really think it's lab shopping. I think it's simply if you run these things as much as we have, you'll realize the error bars are enormous on the plates. And so if you try to get something that PCR is known to have very, very tight CVs, like you can get it to be highly reproducible, but that doesn't help you if the gold standard is a moving target. It's very hard to get our system to give you a CFU per gram that is calibrated to every single plate type out there, every temperature that's applied. And by the way, once in a while, if you throw on the wrong microbe, it melts the plates. Like there's a lot of curveballs that get thrown at you. So I would actually encourage folks to listen to, there's someone in Florida at, what's her name? Ginny, I think. She's on the CanMed podcast. She did this really thorough study where she looked at multiple different plate types, very similar to what we did in our paper. Then she also did one more dimension, is she looked at 24 hours, 48 hours, like 72 hours, 96 hours, even out into hundreds of hours. And she saw that on most plate types, the colony count didn't plateau until like after 72 hours. So if you're counting these at 24 or 48 hours, you're getting a snapshot of the initial growth spurt, but you're not getting all the colonies. And I suspect this may have not been noticed in food as much as it is in dried products. Like you'll notice in the food industry, when they dry things, it takes, oh, there you go. Yeah, Jenny Curry. When you dry things, the microbes kind of go into a sporulation state. And so they don't snap back into doubling times that you would expect from liquid culture. Like liquid culture, you can measure the doubling time of E. coli is like every 30 minutes. Vibrio is really fast. Vibrio is like 20 minutes. But if you dry these things and then rehydrate them, they're not going to like jump out of the gate doing 30-minute doublings. They're going to take time to resuscitate, correct any genomic errors with the DNA repair process, and then they're going to start going and growing. And so there's a delay when you ever have dry products. They always recommend longer growth times because it takes longer for these cells to reboot. I think she's seeing that in her work, is that if you she's handling a lot of dry flowers, and if you run them out, the plating doesn't actually saturate in colony number until you're out past like 5 days. So that's a concern because no one's doing that. And everyone's asking PCR to step into place, give you an answer faster, but benchmark it to a system that's kind of sitting on thin ice, if you will. I think she has some work in there as well about different growth conditions as well. And so now that's one side of the table that varies. Now PCR isn't completely variable free. Let's be honest about that. We've all seen COVID go fucking sideways. People using PCR the wrong way, right? And
we did a whole episode on that with you and Aaron Newkirk. If people want to go back and check that out.
Oh yeah, yeah. So and that is that was criminal what happened, and that they got away under the guise of emergency, that they didn't have to have internal controls, and so like every PCR reaction we have has to amplify something from the plant. And if that doesn't amplify, you didn't isolate DNA, and the test is null and void. You need to show that you got DNA and you did the prep. And that's what the internal control's for. In the case of COVID testing, that's usually a human gene. So you can show that, all right, you didn't just swab your dog and send in the test. Like this thing actually hit a human, and we can count the number of human cells that are there, so that when we get a viral number, we know whether it came from 1 million cells or from 10 cells. A lot of virus and 10 cells, big problem. A lot of virus, tons of, millions of human cells, not a problem, right? This ratio is really important. So anyway, some of the things, the challenges that PCR is facing on this is that, okay, everyone's that's in the marketplace going to have different assays. That's fine. We need that. We need competition. They should all have internal controls that at least show that the plant purified correctly. Some do, some don't. And then the lysis side of this is really important. Some techniques out there are just doing a boil prep, where you just take the cannabis material and boil it and pray that there aren't terpenes and things in there that might inhibit your polymerases. There usually are. We prefer to go through a DNA prep to make sure all that stuff's out of the picture, and then we're only amplifying DNA, and we don't have these boiled samples that have all of these contaminants in there affecting the PCR. Because the plant chemotype varies tremendously sample to sample, and we have to purify that stuff, those variables away. So we don't like the boil prep stuff for that reason, because there can be all types of inhibitors that can happen there. And then the DNA prep's got to be really tortured to make sure you're lysing open all the fungi. We found that for the first few years we were in the marketplace, we weren't lysing open some cannabis species. We never noticed that because Candida isn't found on cannabis frequently, but it was used in a standard that came into the marketplace later from NSI. And we just constantly weren't hitting it, and we couldn't figure out what it was. And then we realized, okay, we're not lysing this particular microbe open because we trained this stuff on microbes we had encountered on the plant. And so we changed some things there to have a much more aggressive lysis to get those things cracked open. So long story short, there's a lot of nuance in how to do PCR correctly, but I do think it's the right approach because it's more universal and more accurate, and that it can speciate, good things from bad things. Yes, there's a propensity that it can overcall things like we saw in COVID. So we got to be aware of that. It should always be trained against, we got to get in the ballpark of the CFUs, but we have to always know that there is no one CFU answer that's right. It's very dependent on the plating systems, but you don't want to be orders of magnitude off from what you're seeing from the plates. So there does need to be a little bit of a sanity check there on, okay, we're not like 100-fold off from what you might see from plates. But it's unlikely we're ever going to get this to agree 100, like within a log scale of all plates. The plates themselves vary by that much. And that's what that intro paper really hit on, is that the plates are varying tremendously just on different temperatures. And that's just one variable that's at play in the whole plating arena.
We ran into issues with this. We've had a couple of times where we've had false positives for aspergillus and couldn't trace it back to any of our ingredients or anything like that with some of our edible products. And then we've also had edible products where some of the ingredients we're testing at, below cannabis or over cannabis thresholds, but under food production thresholds, where it was completely legal in terms of USDA agricultural standards in terms of what was okay for food production, but not okay for cannabis production. We've also run into that with aspergillus in particular, making it cannabis edibles, where all the ingredients tested fine, but they combined, some of them did not, which was very strange.
That is very strange, actually, because usually the kitchen is not going to introduce that, or it's rare that it would. I mean, I can see
especially when we clean with UVC lights at night.
Yeah, I can see like E. coli or something else, but like it's rare you get aspergillus. Aspergillus is usually coming out of the plant as it being, and if you've extracted it already, I would be, that's odd.
We also have some very bizarre
fail rate on MIPS. Our MIP failure rate on aspergillus, from what we've seen, is for what we've heard from our clients, is very, very low. It's usually on dried flower that shows up.
So the worst one I've had testing wise when it comes to cannabis products, and this one we just completely gave up on, was anyone that's gone to Africa knows there's a whole bunch of different tubers that are alleged sexual enhancement, have sexual enhancement attributes to them. I'll leave it at that without getting into specifics. So we tried to find a source of some of these different tubers that we wanted to use for a product that we were going to attempt to bring to market in the United States. And none of them we could get any kind of passing for products in terms of even import to the country, better yet, as far as, actual application of cannabis, because we would have had to like boil, sterilize everything the moment we got it before we'd ever pass testing, because a lot of these tuber crops coming out of the soil, just forget it. Like, don't even try to pass cannabis testing, because if it touched the outside of the soil, it'll never pass testing. And we tried and couldn't get anything to pass testing in internal, R&D batches.
What was it failing for? Was it
everything? Everything across. I could show you some comical failed tests, but it was false positiving for. I mean, we there's no way it wasn't a false positive, but it was specifically with this one group of tubers, this one genus of tubers.
Do you know if they were plating it or using PCR to sort it out?
I think they're plating everything, but I'm not 100% sure. All I know is we tried about 5 different vendors and 3 different labs, and we just gave up on the product.
Oh, that's a bummer. Yeah. All right. That's interesting. Yeah, I don't know what's going on there, but there is something that we're putting in place this year as an option for some of the labs that I think should be really helpful for the communities. That we've signed this deal with a company that built software tools for calling qPCR curves in the COVID pandemic. Okay, so it's like an AI platform that looks at like the slope of the curve and runs through all the logic that I was mentioning before, that like you have to have an internal control that lights up between these CT values. If it's not there, you got to flag this as red, not as green. And then if the curve fits, if the target curve fits within a certain window, then it's a green. It's basically a red, yellow, green system, so people can get a plate of qPCR done, and then have the software just tell them, okay, these wells failed, these ones passed, these ones need to be rerun. So there aren't humans involved in like eyeballing all this stuff, just to speed it up. So they've been able to train these things for millions of tests now. And the helpful thing about it is, well, not only is it shown to be more accurate than like human calling and enable the capacity for labs to like if we had dashboards that could show us that, okay, Arizona's hot right now for E. coli, and that's odd. That stands out next to the neighboring states. What's going on there? Right now, a lot of that information is kind of distributed, and it'd be helpful to aggregate so that the community could basically see that, okay, we're an anomaly right now. Like we're getting really high rates, and all the labs around us aren't. What's going on? We have to figure out a way to do it with the right privacy and such that there isn't, people want to contribute to this, so there isn't like finger pointing going on. But I suspect we could do it state by state, and that would at least give people enough resolution to know that, okay, why Oregon shouldn't be, hotter for this microbe than another state, and maybe there's introduced into that environment. But those are the types of things we're looking forward to, because I think that would help find more cases like what Steve and Chris found. That, okay, this is an anomaly, and it was only recognized because they had expertise in going on. They're able to I mean, I wish we had more of that, frankly, in all the other aspects of it, because I feel like this whole Valley thing got like fakely blamed on cannabis because we didn't have good tools to aggregate what was going on in the marketplace. And so they just said, oh, yeah, it's got to be THC, even though they found it in nicotine pens. And I've seen some compelling evidence that may have been an earlier wave of a coronavirus, because it had like a Gompertz curve to it. It came in and went out at the rate that you would see a pandemic come in and go out, and it had ground glass opacities and a lot of the same symptoms, predominantly hitting the young. But that was how they diagnosed it. They didn't. There was an age designation to the diagnosis. So if you were old and had it, they didn't call it a valley. That's very tentative and obviously hypothetical, but we were unprepared as an industry to aggregate that information and try and find those patterns.
Are you talking about valley floor disease?
No, a valley was the vape disease they blamed on vitamin E acetate. Which this is just before the coronavirus pandemic.
Yeah, I remember that. And in fact, I full disclosure, I use vitamin E acetate with edibles. So
it's been used.
I thought it was kind of strange.
It's been used for a decade in vape pens before this. So why did it suddenly like show up now? And that's why I didn't. To me, I thought it was very. And I saw a couple people who made really good pens get harassed in the industry as being like snake oil salesmen for having vitamin E acetate, products, but they had been using those things for years before this thing came and went. So, what was new about that? And we would understand. I think we'd be able to sort that out much faster. I no longer trust the CDC to figure that problem out anymore. they've just proven themselves to be wholly incompetent throughout COVID. That if our solution to finding these types of manufacturing problems in the cannabis space is to hope the CDC comes forward with vitamin E acetate, is like we're just setting ourselves up to get railroaded.
Oh yeah, it's right up there with Stop the Spread. Remember that one?
Yeah.
Anyways, we covered that in a whole episode. If you guys want to check it out, episode 251. We did an entire episode about coronaviruses and cannabis and side effects from various control methods without triggering the algorithm there. We'll leave it at that.
Right.
One of the other things I wanted to mention is, and something I wanted to talk to you just because I feel like you could help put the nail in the coffin of this. There's a green powder out now, which as far as I can tell is just maltodextrin with green dye being sold for hot latent viroid. And I'd love for you to take 2 minutes and just talk about how ridiculous that is.
Don't know anything about it.
Have you seen it yet?
Oh, no. What is it?
There's a green HLVD powder. Let me see if I can find it real fast. But there's someone selling a green powder, which looks like it's just, I'll find it here. Hold on a second. But it looks like maltodextrin with green food dye in it. I'll be frankly honest with you.
Probably a PCR inhibitor.
Is it?
I was kind of curious
because people are advertising this as a HP. Hold on, let me find the person that sent it to me. Here it is. It's just the most ridiculous shit that I've seen. But I wanted you, because you're someone who's actually tests the DNA of these types of viruses and things like that, and other issues with plants. Like, if you could please touch on how just ridiculously stupid something like that is in terms of, there's no one's invented a powder that suddenly cures HLVD.
Well, no,
certainly not one that you and I have not heard of.
Yeah, nothing in peer-reviewed literature, but I don't. Hey, I'm open-minded, right? If someone's figured this out and it works, I don't want to like blindly, shit on it. But
this was very clearly snake oil. That's why.
Right. I'm happy to give it a shot. Actually, I have an HLV plant that I am sadly.
I have for something for you to try then. So Chris Trump is having really good luck with mosaic viruses and nitrates with treating with IMO collections. Liquid IMO.
Yeah, this thing I've been struggling to keep alive just so we have something to test, and which is a bummer because I really can't grow anything else because it's just, this thing's a nightmare. I have to like kind of constrain myself to having one disease plant in my life. But I need it for just like getting access to materials so I can keep honing these tests. But yeah, if there's something that can possibly, we have a few people who've got cuttings from this thing. They're trying to tissue culture it out. Experts in that type of stuff that we're going to, PCR it throughout that process to see how well is the tissue culture eradicating this thing. But if they think they found something like that would lower it, well, I'm happy to give it a shot and see if I can certainly see if the CTs move around. I'm somewhat skeptical though. These plants look like they are on the brink of life. I mean, they're totally wrecked.
Yeah, I can't seem to find it now. It was from Matthew Gates. He had a post about it the other day about how this was very obviously snake oil, and I agreed with him when I read the thing on it. But they didn't have any paperwork to go with it. They didn't have anything really to prove their claim about whether or not it actually did anything against the powder. Why can I not find this now? It was right here.
Yeah,
anyways, I'll find it later.
We probably had a chat about this before. I'm trying to remember if we went over HLV in the past feed, but if not, I'll just reiterate the one thing that we've noticed in that kind of testing is that when plants have it, they're screaming hot. I mean, the PCR signals, we don't miss them. They're like the CTs are in the 20s. There have been, a lot of some chatter in the field about people getting down to single copy, or 4 copies of testing for this. I think that's a little bit misguided, that you're more likely to be chasing false positives when you do that. Now, that's fine if you want to throw out clones, because clones don't cost very much. But if it's your mother plant, I would never go off of a late CT on an HLV test. If it's over 30, rerun it, because it's probably an artifact. They should be screaming hot. And if it's consistently over 30, you probably have something biologically very interesting there, because most plants don't do that. It might have some immune system that is actually fighting this thing quite effectively, and you might want to breed that thing.
My question was, is it possible that people could have the virus present, but that it's not at a viral load, that it's actually affecting anything yield-wise? Because we certainly see that with mosaic virus. And I didn't even realize that until, and I told you about this last time you were on the show. I was at a facility last year, and I can privately give you the contact info of them if you're really interested research wise. But they had a line at this facility, and they had 4 different grow rooms that were in flower, and one of them, each one had different light settings, but they're all from the same mother stock. And only one of them had a light spectrum that was triggering the mosaic virus, whereas the other 3 rooms had cuts off the same mom that had no mosaic virus.
That's interesting. Yeah.
But it was the light spectrum that was the only variation. All the nutrients were identical. They were solely testing light differential as the variable.
Yeah, no, we'd love to talk to them. That might shed some light, no pun intended, on some interesting biology that's going on.
Yeah, because the light spectrum is going to affect the expression in a way that's quantifiable that you could use for testing purposes or some other way to narrow the shit down. So that I've never observed, other than that one facility, never observed the light spectrum being a factor in virus expression for these types of anything, really.
No,
so that was really bizarre.
Because if you take an HIV positive plant in veg, it's hard to see the symptoms. To 1212, yeah, so there's something about something going on in the flowering process that is I think so I wouldn't be surprised if like the light spectrum has a similar artifact as well. That's very interesting. But you raise a good point about asymptomatic. Like these are latent viroids. So what happens? Do all plants actually succumb to having dudding and these chlorotic leaves when this thing is present? I don't know that we have that answer. There could be asymptomatic carriers of this thing. In which case, if it doesn't affect yield, what's the risk of that other than to your other plants that aren't resistant to it, right? Resistance, exactly. I'm using in air quotes because it's not really resistance, but you know what I'm saying. If you're an asymptomatic carrier and it doesn't affect your yield, you're only a threat to other plants that aren't as robust as you are. So that's I don't know. I'm just a little bit nervous about going into PCR mania like we did with COVID and calling the shit out of all these ancient genetics in the field because we think we understand this viroid when we don't really understand it yet.
Oh yeah. I mean, shit, any of the chem dogs, you can get that weird streaky patterning that looks almost identical to mosaic virus. And, does that go back to attribute to that, or is it just a similar expression because that expression can be kind of expressed similarly? So many different ways that,
they could have other mitoviruses as well. We don't really know what the mitoviruses are doing, but we found mitoviruses in the Jamaican lion genome when we sequenced it, and they're kind of just parked there. We don't know what to do about them. But these are mitochondrial viruses that are oftentimes only brought out when the plant's under stress. So,
or can they be used like that Aspergillus strain as to neuter the pathogen so that if the plant's already infected with something, so it can't get infected with the other? Like, maybe they end up being some of the best benefits that we have to preventing some of this shit. That's the thing that's so cool about some of the stuff that you do and the science that you work on, is that this is all the things that we still have question marks on. And it's so cool that we're in the period of discovery. And
it is important to step back and realize that viruses have been brought to public consciousness through Fauci-like fear, right? Humans always think virus bad, but if these things have been evolving with us since the beginning of time, there's probably beneficial roles for them we don't understand, right? I mean, not everything can be parasite. In fact, parasitism is probably less effective than mutualism. I mean, the vast majority of interactions in life are more symbiotic than they are parasitic, and we just don't probably fully appreciate this. But so just because something is RNA and it replicates, and particularly when you start getting to things that are like asymptomatic and latent, you have to start asking, okay, the disease is really showing itself when the person's immune system goes down. But it's otherwise not a nuisance, right? So maybe we've yet to figure out what purpose they play for the survival of the population. Because there may in fact be a benefit for them that we don't fully appreciate yet. So I don't know. I think like the virus fear porn that we have going on in human biology is really very pharmaceutically driven. That we need to panic people over these viruses and treat them and inject them and all these things. But in biology, I think we probably have to step back and realize it's a lot more complicated than that. And there may be a role for these things sharing RNAs. I mean, we certainly don't look at exosomes as being like this infectious, scary thing. And we know that these exosomes these are like, lipids that your cells butt off. And there's usually microRNAs and other messengers in those things. And they're communicating, they think this is like an internet of the cells, is to share these exosomes around. Okay. So that's not too different from what a virus is, right? A virus is just a protein code version of sharing some RNA around. And we've been drawn to the ones that are pathogenic out of a medical necessity, but I'm sure there's plenty of these RNAs getting shared that are either mutualistic or not necessarily pathogenic, that are important in human biology.
Great examples of that would be wasps need a virus for their babies to fully develop properly. Otherwise, they don't form properly and create the right shape to be an adult wasp.
There you go.
Actually, there's a really cool paper out not that long ago, maybe the last month or so, on the virus component of the development of wasp larvae. But if you're looking for something else down that exact topic that you're talking about, there's another one on bees as well. There's a really interesting study that was put out in the last year on whether or not you use antifungal agents on your garden.
Yeah, Paul Stammet was involved in some of that work.
Yeah.
Right?
Yep. On how the antifungal agents that we're applying to stuff with pollinators are being brought back to the hive, and they're killing the fungi that the larvae need to digest, the bee larvae need to digest their different sugars and things, so that the bee larvae are not actually able to digest them anymore. And they're ending up with 25% of the surviving larvae compared to 100% without it. So,
yeah, well, I mean, if you just look at the human genome, they're like over 50% of it is these ancient viral elements, like these lines, signs, alus, right? These long terminal repeats. So, and there's a whole other class of them that are retro elements. I think like 8% of the genome. So there's a portion of the human genome, a large portion of it, that actually codes for these viral elements, and some of them are still active. So, like, this is not necessarily something that we can't take this blowing the bubble, all virus bad approach. We have to kind of step back and recognize there may be a place for HLV in the evolution of cannabis, and a full-on eradication program like a zero HLV program is likely to be just as economically destructive as zero COVID.
I couldn't agree more. And that's really where I was wondering, and why I asked the question about, hey, is it possible that it just has to reach a certain viral load before these yield effects are being noticed, like it is with Mosaic, where you can have it fully latent and completely, while genetically detectable, not expressing in the plant unless conditions are met? Or, because I think honestly, at the end of the day, and again, I have no quantifiable evidence to prove this, but I do feel like, given that how long we've all been growing cannabis and how new of a problem HLV seems to be, that it really is something where if it's below a certain viral threshold, and this is, again, my personal opinion, it's not going to be a huge problem. The same way that we see with Mosaic, where if you have healthy soil, it's biodiverse, the plant's healthy, it's a good environment, it's going to suppress it 9 times out of 10 and not be affected.
Yeah.
That's my personal opinion.
I think it's a fair assumption in that, in absence of any other hard data, that's what we see in a lot of other biological systems. I mean, we've seen it in, I don't want to get your podcast banned here, but we've seen it in other viruses that have recently hit humans, that vitamin D and these other aspects that support your immune system. If you have a weakened immune system, you're going to get hit by these things. But
it was a 97% reduced chance of infection with vitamin D above, I forget what the threshold was.
I don't even quote those things
something
anymore.
Crazy. It was something crazy. It was something crazy.
Yeah, that's one of the trigger words on like Twitter and Instagram is vitamin D. So I kind of stayed away from it. But I think everyone's gotten the message by now that's actually really pivotal to have your immune system in gear. And without it, you're going to be more prone to this. I would imagine in the plants, not that you feed plants vitamin D, but I think people get the analogy that the plant has an immune system as well. And in the case of the Vibroid, I think we need to do more work on like the RNases, because this thing seems to have evolved a way to evade a lot of the other RNAs that are RNases, or these enzymes that destroy double-stranded RNAs. And you'll notice hop latent Vibroid is double-stranded for really short segments. It's usually less than like 20 or 30 base stretches that are double-stranded, and then there's bubbles everywhere in the genome, and it's circular. And because of this, it's been more resistant to some of these RNases, particularly RNase 3 is the one that usually takes these things out. But there is an RNase 3 gene in Arabidopsis. There's one in cannabis, and we may find some variations in those pathways that predict which plants like tolerate this thing better than others. So I think there's a whole field of research there to sort out what we've got going on with the innate immune system in the plant.
We had a question from chat. It says, is there a potential for viruses to be transferred through mycelial networks?
Yes. That's been documented with hop stunt viroid, I think. Actually, it's gone through, they've documented through the mycelium. And I think it was in hops, actually. I'll have to take up the paper for you guys, because that's what got me thinking, if hop stunt viroid can do it, why couldn't hop latent viroid do it?
That makes a hell of a lot of sense, because they're cousins.
Yeah, they're very small, right? When you get into viroids, they're like under 500 bases, so they're tiny. They don't need a protein coat to infect. They're small enough to like get through cells without having a transfection tool, if you will. I mean, they're a very important part of this terroir virus kind of theory that's been hotly debated in the C19 space. Which there's a whole class of scientists out there that just don't believe the virus has ever been properly isolated, and are concerned that the EMs aren't done correctly, and so on. I don't subscribe to that from what I've read, but there is, I think the viroids kind of throw a big curveball in Koch's postulates that they need, is that they're always looking for this protein coat to prove a virus exists. And, people start raising their hands. Well, what about the viroids? They don't have any. And we can clearly show you can take a viroid and move it from one plant to another with mechanical transfer. There's no ethical violation doing this, like we have trying to study viruses in humans. And so, Koch's postulates pretty easily obtained with viroids in plants. And the same thing is kind of being brought about prions as well, because prions aren't replicative like viroids are. Viroids have a mechanism to replicate themselves. Prions are proteins that don't necessarily replicate themselves, but they're infectious. And there's disease that you can transfer from one organism to another. So it's thrown a whole nother curveball into Koch's postulates. But you have to remember, Koch's was a postulate that was, again, back when plating was around. It needs to be updated with the tools that can measure these new things, and we've got to keep our mind, I think, current on this and not get stuck in sort of a Luddite type of definition of these things. But anyway, I think that's very possible that they're going through mycelium, and so if they're in the same root bed, you may not have any sign of mechanical transfer. And you may have the flies and the insects completely under control, and then see it pop up in another plant that's in the same mycelium connection, and that would probably be the hardest evidence of it. But anyway, if anyone has an example of that, contact us, because we're very curious as whether we can document that, and how we'd go about doing that. We have to kind of brainstorm with people who have access to that type of scenario. But I owe you guys a paper on that for stunt viroid that got us on that topic.
If you guys do have a hop latent viroid or suspected hop latent viroid tissue sample that you can send him, you can reach him through Kevin McKernan 73 on Instagram, or you can reach him directly over at medicinalgenomics.com, and I'm sure he'll get a hold of you and get with you on that. I did have a question on, is there anything like prions in plants? For those of you that don't know what a prion is, prions are misshapen proteins normally found in the brain that can cause horrific diseases, unparalleled compared to just about anything other than maybe like a hemorrhagic disease, in terms of it rots your brain and hijacks your brain and makes you someone else before it takes you. It is a very horrible disease, but or classification. You have Gene Fitzgerald disease. You have mad cow disease. You have the human version of that, and a couple of others that make up that classification. There's one at the cannibals they identified in Africa as well. That was a different one. Anyways, is there anything in plants that's like that same kind of thing, like a prion, where it's a misshapen thing that can be transferred that way?
That's a good question. I am not familiar with that, although, you can quickly Google and find plant protein behaves like a prion. I don't know that necessarily manifests itself as a prion in the plant, because there isn't a nervous system necessarily for it to bioaccumulate in per se, but it maybe affects some other tissue. But yeah, that's a good question. I can't see why it wouldn't be the case. I don't think there's anything about prions. I'll ask Stephanie Sneff about that. I'm on a call with her once a week. She may have some info on that because she's been going deep onto prions lately.
Evolutionarily, it seems like it would be advantageous for it to evolve a similar mechanism, but especially if it could transfer through mycelial networks.
Yeah, I mean, if it's a way, if it becomes kind of a bio, if you will, a way to ward off pests. I wonder if that's kind of like a poison of some sort. But it's certainly that's a rabbit hole you just sent me down. I'm gonna lose sleep.
Like the ultimate pesticide. Denatures their proteins.
Oh man.
There you heard it first. Groundbreaking science. No, I'm just kidding. But
plants can take up prions from the soil.
That I did know.
Animals.
Yeah.
Okay.
That I did know.
They make any of their own is a really good question. All right.
It's either that
next time we get together, we'll have to do a deep dive on.
That would be cool because it's either that or plants evolved a mechanism to prevent that, the way that sharks do with cancer and things like that. So
yeah. Hey, it's Shark Week soon. Got to celebrate that.
Right? Yeah, it was next week, I think it is. So
yeah.
Well, is there anything else you wanted to touch on? I know you didn't have a ton of time tonight. I don't want to take too much of your time. I do appreciate you taking the time kind of last minute to join us this week.
No, thank you very much for giving me the time with you guys. I always learn something from you. Always ask me questions I can't answer. I love that.
Well, I always love speaking with you, and I do appreciate how much support you give our community and the help and assistance you've given me over the years, and I always try to support you any way I can. Again, if you haven't checked it out, really great white paper. Check it out over at Biocontrol Agents and Their Influence on Cannabis Testing Space. And his other paper he put out, or his co-author on, is pathogenic Enterobacteria ACA, a requirement multiple culture temperatures and for detection in cannabis sativa. They're great options for you guys to read if you're looking to learn more about testing and testing regimens, the way cannabis is tested, false positives, things like that. We did have one last question in chat. It looks like, can Kevin explain the type of testing they're using?
Oh, so the testing we're using is PCR based at the moment. So we utilize polymerase chain reaction to target, for E. coli and Aspergillus, it's very species specific and targeted. It only gets those organisms, and we can have really clear inclusion and exclusion criteria where we test like 50 organisms it should hit and 50 it shouldn't, and demonstrate that it's that the primers are very, very specific. Now, when you get into the total testing, it gets a little bit more vague in that we have to then target 16S amplification. So all bacteria have a 16S ribosomal DNA element. That's how we can go after those. Now, if you choose wisely in that area, you can go after a class of bacteria that generally is in the total aerobic category and avoid the anaerobes because they tend to have 16S sequences of more similarity. So we've designed primers that specifically amplify those. For fungi, there's another region that fungi have conserved known as the ITS region, which is an internal transcribed spacer, also a ribosomal DNA element. But that is just better to target those. If you need, in order to target the ITS region, plants also have ITS. So you have to design primers that target the fungi, don't hit the plants. Now, there's a couple other aspects. I'm probably going deeper than you want, but others have asked these questions. I've seen them in blogs, so I'll just address it out of the gate here. These regions in the genomes are usually variable in their copy number. Okay, so bacteria tend to have maybe 6 to 10 16S regions, and fungi can have like 20 to 100 ITS regions. In fact, psilocybe, we think, has like 36. We did a lot of psilocybe sequencing on that front. So a lot of people are like, whoa, wait a minute, you got a lot of copies. This is how you're going to really figure out CFUs if there's a lot of copies here. But you have to remember, this is like, I'd rather be arguing over 6 to 10 copies than 0 or 1, which is what we're getting on the growth side. Like some things just don't grow at all. If you see some of the pictures in that paper at certain temperatures, a 0-1 problem is a real problem. A 6 to 10 is a calibration problem, and it gives you more sensitivity. We would rather have higher sensitivity so we can get sub-CFU sensitivity, if you will. If there's six targets per genome, the genome can get obliterated, half of it can be missing, you can still pick it up, right? So the ITS and the 16S copy number, yes, it adds a variable in PCR calculations from CFUs to CT scores. However, it tends to give us an overcalling propensity that we can pick up things that are sub-CFU, and you can't do that with plating. That can be calibrated a little bit, but there are bigger variables at play than that log scale that's in flux. The bigger variables, as you'll see from the papers we put forward, are some things just don't grow at all on some plates, and so you can never capture them. Anyway, a bit of a deep dive on what we're doing there to pick these up. How do we validate these things is we tend to do sequencing. Right now, we're doing a lot of nanopore sequencing, but we've in the past did a lot of the stuff on Illumina, where we would do whole genome sequencing or ITS and 16S sequencing, just to confirm that every time we get a positive PCR result, that in that mixture, we can speciate everything that's there by sequencing it to know that, okay, that you had a positive TAC signal on PCR. If you sequence it, what do you see? Do you see any fungi coming through? And do you see any off-target material coming through? And by sequencing those amplicons, you get to resolve what's contributing to the PCR signal. That's something you can't really do on plating. You can try and get a MALDI-TOF involved to run around and mass spec the colonies, but you only get the things that do grow, not the things that don't grow on that given thing. And the MALDI-TOFs don't do a very good job speciating, actually. In fact, there's papers out there looking at MALDI-TOF and Aspergillus, and it's a total train wreck. You can't really sort them out with the MALDI-TOF. So the real form of speciating these things is the DNA sequencing in these loci. These are the taxonomic things that they hang things on the tree, is 16S and ITS, and that's kind of the way to speciate things. So we're sticking with kind of gold standard sequencing to understand what the PCR assays are in fact picking up.
Awesome. The one last thing I did want to mention here, if you guys really like his information, you can check it out over at medicinalgenomics.com. We also have, he has a whole bunch, oh, I'm sorry, I said that backwards. He's been on the show 4 or 5 times before. You can check out his previous episodes. You can also check out the wonderful event that he runs called MedCan, where he takes all the people that he learned, CanMed. Yeah. Sorry. It's been one of those days, man. The whole week has been kind of insane.
Yeah. All the videos are up now, by the way, from 2022.
Yeah, that's what I was just going to mention. If you guys can check out all the CanMed videos over on his website, I'll pop up here in a second. But there's a ton of just incredible talks that are highly beyond anything else that's out there on the market in terms of detail and specificity and all kinds of things. And certainly, Dr. Mycelion and all kinds of other people here that you're not going to see anywhere else hardly in terms of giving these long format talks. So definitely one of the best conferences that you can check out. He also has his previous years on the website as well, which is a wonderful resource. If you haven't checked them out, you really should.
5 years maybe now that are on the web, all for free. And I think the news is out that we are moving this to Florida next year.
Nice.
And we'll keep you apprised of that. I think there's some announcement coming out on where it's going to be, but it's a fun location. So it'll be on the East Coast and should be easy to get to from everyone from Europe.
Wonderful. Definitely looking forward to trying to make it out there this year for the next one.
Oh yeah, it'll be great. Got to get the Oklahoma folks down there.
Well, it's definitely one of my favorite things to watch. I was actually watching it earlier this week, so definitely one of my favorites. Well, thank you so much for your time. Do you want to tell everybody a little bit about how they can find you and some of the other services that you provide? Anyone can find him over at Kevin_McKernan_73 on Instagram. And then I'll throw your website up here on the screen.
Yeah, website's probably the easiest at Medicinal Genomics. My Instagram is a mixture of probably unrelated things to cannabis sometimes, but at least lately it has been. Likewise, I have a Twitter account as well, which tends to be a little bit more focused on. Lately, it's been a little bit more about C19, but occasionally there's some psilocybe and cannabis stuff thrown in there. But most of the cannabis and psilocybe stuff you'll find at Medicinal Genomics and the other site you mentioned.
Awesome. Well, I very much appreciate your time. You have a wonderful podcast as well that you guys also put out that you can find also through their website as well. And the CanMed podcast. If you guys are looking for more highly detailed cannabis science talks, definitely check that out. And thanks for taking the time to come join us today.
Excellent. Thank you so much as well, Steve.
Really appreciate you. Thank you.
All right. Cheers.
Have a good evening.
Take care.
Again, you guys can check them out at medicinalgenomics.com. over at CanMed podcast. If you want to hear some of the talks or their videos, you can check that out as well. Again, really wonderful resource of these higher level talks that goes into the science a hell of a lot more than most of the other sources out there on the internet. You can also find us over at myself at apcannonoots.com. You can also find us at aptestkits.com if you want a full list of test kits. We don't mention that very often, but I had that up earlier today for talking with somebody else. Also thepestclass.com. If you guys haven't checked it out already, we do have that as well. If you're looking for a full-length pest course, I will be finishing up adding the last couple of slides to that. We do have a full day's worth of content on there, and I do have about another half a day's worth of content to finish editing. That's all edited up, or it's chopped up. I just got to finish editing. And then you can also find us over at apmjclass.com. Oh, I thought I had it up here. One moment, guys. I hit the wrong button. There we go. Now it's working. You can find us at apmjclass.com if you're looking for a full-length aquaponic cannabis course. We do cover all aspects of aquaponic cannabis content. I have started uploading some of the new videos on that. If you are a student, be sure to check it out. We do have quite a few new slides and new talks going up on the course. So if you're doing that, and then I just finished it putting together all the content for the next course that we're putting out, which is the microbe and mineral course. So if you're looking for aquaponic mineral and microbial classes, specifically on those topics, and you have the rest of it down, or you're a vegetable producer and don't really care about the cannabis aspect, we will have a shorter version of that covers all the important aspects of that across all different types of crops with different considerations for different types of crops considered. On the slides, it talks about different aspects of root crops and flowering crops and lettuce crops and things like that. So we should check that out. We will have that available here in the next 30 to 45 days. I do have a little bit of travel here coming up, but we should have that out here before the end of August. So be sure to check that out as well. That'll be up on the website. I've been working pretty swiftly on the book as well each night, so we can check those out. Here in the future, I have a whole long aquaponic cannabis book that we're putting together. I'm also going to be taking and making a aquaponic vegetable version of that book here once the cannabis version's finished. But we're already at 197 pages, and I'm not even a third of the way down the book in terms of fleshing out with pictures and some of the other awesome detail that I want. So it's going to end up being 400 or 500 pages, I think, when we're done. So certainly the most extensive aquaponic book that's been put out there for sure. But that's chugging along, and be sure to look for aquaponic cannabis edibles coming out here soon in Oklahoma. Hopefully in August, we'll have those through final testing and on the market. If not, September, you'll be able to find new cannabis edibles in Oklahoma. Be sure to check that out. We also have at the end of the month, I do believe it's next weekend. Let me double check. Yep, next weekend is the Supernatural Conference. I'll be speaking there along with Cannabis. We did an awesome episode with her a couple of weeks ago. Chris Trump, great friend of the show. Jace Riviera, Ben Acadia, Dan Kittredge, a whole bunch of other awesome people. Be sure to check that out. Last weekend of October. Sorry, it's been a really long week and a long day, and my brain hurts. A migraine I cannot get rid of today. Anyways, we'll see you guys there the last week of July, the last weekend. We also have an after party at Ace of Eights. I'll be trying to there as much as possible. And then what else do we have going on? Oh, Myceliate the festival. Myceliate festival. All right, so I'll also be speaking out here with Chris Trump as well, good homie, August 3rd, 19th, 20th, and 21st. Up in Washington State. So be sure to check that out. We have a ton of awesome speakers, and I'm really excited about that festival as well. Be nice to get up into the redwoods and off of the scorching hot 40 degrees Celsius temperatures that we've had all week. Actually, it was 44 here yesterday Celsius for those of you in other areas. So we'll, any excuse to get out of Oklahoma in August is a good one. And certainly when you can go hang out with good friends and have a wonderful time in the forest, it really can't be beat. So be sure to check that out. I'll be talking about how to build your home aquaponic systems. Do I still bump into Harley Smith at the airport? No, but him and I, somebody asked in chat, do I still bump into Harley Smith at the airport? No, but him and I have this weird thing. I bumped into him probably a dozen times in airports where we're both going to different events or the same event. And it's kind of this little weird comic joke that we have going on where, I always try to buy him a drink anytime I see him in an airport bar. So always fun. Anyways, thanks everybody for watching. It is, this was kind of a last minute thing. I really wanted to do a talk about his video on the aspergillus paper, and we just could not get to where Thursdays were going to work. So we decided, screw it, we'll just do it on Wednesday. So that's how we had the episode today. So we may or may not have an episode tomorrow. I'm going to try and have one tomorrow, but if our guest is unable to make it, then we're going to switch it around because it was a maybe for tomorrow. So that was kind of part of the reason for doing it today with Kevin instead. So. And then next week, there's a good chance we're not going to have an episode next week. We have the Regen conference in town. It's going to be a lot of people in town, and probably just won't have time to do an episode next week. So if I don't have an episode before Thursday, we probably won't have one next week, just as a heads up. But we will be back again the following week. And then the week after that, we'll have one after that. And then the 18th of August, we will also not have a show. As I will be traveling and flying that day. So. I don't think so. Did he? I don't want to say that on air because I don't know if it's true or not. Hold on a second. I need to look into something. I don't think so. I'm pretty sure I just saw him. One second. I'm sorry, guys. I don't mean to give you guys dead air if you're listening to the audio version, but someone said something and I don't want to say the wrong thing. No, that's not true. Whoever said that in chat, that's not true. But I can verify at least. So, all right. Well, we'll leave it at that because I certainly don't want to misstate something like that. Anyways. We will certainly see you guys again sometime in the next 7 to 10 days. I'm not quite sure when. But we do have some other cool content. I have a bunch of stuff that's filmed and edited that I just need to finish editing to get up, and you guys are going to enjoy it. And we also have a bunch of other cool events that are in the works for the fall, and possibly some travel. There's a bit of some stuff in the works that I think you guys are going to enjoy. And other than that, if you guys are interested in speaking at the Aquaponic Cannabis Conference, please hit me up. We are looking for 1 or 2 more people for the last couple of slots for that. We do have most of it booked out now. We just have a couple slots left in terms of producers. And then remember, that's the first weekend of November. If you're looking for more aquaponic cannabis information, this will be our 3rd year doing the Aquaponic Cannabis Conference. We're very excited to put it together. We have quite a few awesome speakers this year, and definitely looking forward to hearing the talks for this year as well. All right, guys, thanks a lot for watching, and we'll catch you guys again. You can find us on your favorite podcast app. We are on basically everything at this point: SoundCloud, YouTube, Spotify, iTunes, all the things. And we'll see you guys again next week. Cheers.